In this video, we are going to describe about what is epistasis, the types of epistasis, that is dominant and recessive epistasis, and the particular example where dominant or recessive epistasis can work. So let's start with what is epistasis. So epistasis is a phenomenon in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutation in one or more other genes, respectively termed as modifier genes. Alternatively, what we can say, when a gene for a particular trait is inhibited by another non-allelic gene from expressing the trait, we call the latter as the inhibitory gene, and the phenomenon is known as the epistasis. Epistasis can be of two types. One is known as dominant epistasis, another one is recessive epistasis. In dominant epistasis, a dominant allele at one locus can mask the expression of both alleles of another gene at another locus. But in recessive epistasis, the recessive allele of one gene can cause a masking effect on the expression of alleles of another gene at another locus. Let's take example and understand the details of dominant and recessive epistasis phenomena. So this is particularly a picture where dominant epistasis is explained. As this example, in poultry, white leghorn and plymouth rock are both white. White leghorn has two genes for color and two genes for inhibiting its expression. As we can see here, this capital C is responsible for the color formation, but capital I is present. These are the inhibitory genes for the expression of the color, and that is why they appear as white. But plymouth rock has neither the color gene nor the inhibitory gene. That means it contains small I and small c. So they will eventually produce different gametes, that is capital C capital I, small c small i, small c small I. So upon F1 progeny, that is first filial generation, all these poultry will be white in color because they also contains the inhibitory genes, that is capital I, right? So the F1 will all be white, and in the F2, white and colored chicken appear in the ratio of 13:3. So we can see here that is these are the different gametes, that is the gametes produced from this first filial generation will be capital C capital I, this is the first one, capital C small I second one, small c capital I, this is the third one, and small c small I, that is the fourth one. So we can generate different gametes, and we can explain this in Punnett square, where what we can see that 3 out of 16 will be colored, 3 out of 16, and this 3 will be number 6, number 8 and number 14, because these three that is 6, 8 and 14, these three do not contain the capital I gene, that is the inhibitory gene is not present, but capital C, that is the gene for color formation, is present. That means 6, 8 and 14 will be colored as they do not have the inhibitory genes but have the genes for color. All the others will be white because the inhibitory gene prevents the expression of the gene color, or we can say the genes for color is hypostatic to the inhibitory genes. Let us take another example. In summer squash, 3 types of fruit color appear, that is white, yellow and green. White color is controlled by dominant gene, that is capital W, and yellow color by dominant gene capital G. White is dominant over both yellow and green. The green fruits are produced in recessive condition, that is small g, small w. A cross between plants having white and yellow fruits produce F1 with all white fruits. We can see here these are the all white fruits, right? So intermating of F1 plants produces plant with white, yellow and green color fruits in F2 generation in 12:3:1 ratio. Here W is dominant to capital W is dominant to small w, and epistatic to alleles capital G and small g. Hence, it will mask the expression of capital G or small g allele. Hence, in F2 plants with capital W, capital G, that will be out of 16, 9, and capital W, capital small g, that is 16 out of 3 genotypes will produce white fruit plants with small w, capital G will produce yellow fruit plants, and those with small w, small g genotypes will produce the green fruits. So here we can see that is these are the actually the green fruits, that is only one, and there are some yellow fruits here. These are the yellow fruits because small w is present in both of the cases, that is why it is yellow. These are the all small w cases, and all the others will be white because it contains at least one copy of capital W. So the dominant epistasis for food color in summer squares, the normal dihybrid modified to 12:3:1 in F2 generation. In the previous case it was 13:3, and in this case it is the 12:3:1. Now let us move to the recessive epistasis. The gene for albinism is epistasis to the gene producing color black and agouti. What is albinism? Albinism is the lack of pigment. When the genes for albinism are in homozygous condition, they will not allow pigmentation. As the gene for albinism is recessive, two such genes are necessary to prevent the expression of the genes for color. In human beings, the genes for albinism are recessive to black pigment of the skin, but if two such genes for albinism are present, they prevent expression of the black pigment color in the skin, even negros. So let us take an example here. So black and albino mice when intercrossed, here this is the black and albino mice, the wild type coat color, that is this is the capital A, this one capital A. The wild type coat color capital A, that is agouti color, is dominant to solid color, that is the small a. So if we compare between capital A, small a, if capital A is present, that will be agouti color, and if only small is present, that will be the solid colored fur. However, separate gene is required for pigment production, that is capital C. C is required for the pigment production. A mice with a recessive allele small c is unable to produce pigment and is albino regardless of the allele present at locus A. So all the albino mice will be of three types, that is capital A, small c, one will be capital A, small c, and another will be this one, that is the capital small a, small c, small c. So the C gene is epistatic to the A gene here. So black and white albino mouse when intercrossed produced F1 generation, all of which had agouti color. This is due to the genes supplementing each other. But in F2 generation, 9 will be agouti color, 3 will be black, and 4 are albino. The 9, that is agouti color, will be 1, 2, 3, 4, 5, 7, 9, 10 and 13. Look at this. In 3 cases, that is 6, 8 and 12 are black, and 4 cases, that is 11, 12, 15 and 16 are basically albino. So the ratio becomes 9:3:4. This can also be explained by a picture. So this is the situation where both of the mice are albino, like the previous one, agouti color. I am very sorry, this is the agouti color. This is the capital A, capital C, small a, small c. This one is capital A, small a, capital C, small c. So in this case, 9 will be our agouti color, 3 will be black, and 4 will be albino. So the ratio is 9:3:4. Hope you understand.